As you may all notice, as we're getting ready, I think I'm going to close this tab. Oh, weird to see myself with a delay. I am live streaming our sessions on YouTube so that the videos will be available immediately after class because YouTube archives them immediately. Hopefully that'll get you access quicker if something comes up and you need to miss class. Okay, looks like there's no one in the waiting room, so everybody might be here. Everybody on time, that's awesome. Okay, so hopefully you're all in the right place. Don't know how you would have gotten here otherwise, but this is Bio 1036, Biology of the Cell, and I am Dr. Zelazny. So a little bit about this class, right? This class is designed to prepare you for A&P and for micro. So the big target of the class is to cover some real basics of cell biology. So as in future classes, you dive a little more into the function of cells and how they work with each other, with their environment. You have a basis for that understanding. Also a little intro about me. I'm Dr. Zelazny. I teach this course as well as the entire A&P sequence, including all the various labs. So if you do move into A&P next year, you may see me again, very likely. I'm mainly a bone biologist. I study the evolution and biomechanics of bone, but I've been teaching bio of the cell for a couple of years now, and I've come to love cells, which is a sentence I maybe thought I would never say way back when I was an undergrad. I was more of an ecologist back then. Okay, so welcome. I'm very happy you're all here. So I hope you all got a little scheduling note. You may have gotten here from the Brightspace course, or you may have gotten here from my Google Calendar invite. If you haven't checked, it might be in your spam or something, but I sent everybody a Google Calendar invite to this course that has the Zoom link every day. The Zoom link stays the same every day, but the advantage of using the Google Calendar invite is I've gone through and I've deleted days where we don't have class due to those no contact days. So it may be helpful to go through your calendar as opposed to going through the Brightspace course, unless you already have your own personal calendar where you've crossed out all those sort of sporadic spring break days instead of our usual week. Okay. So moving forward, actually we'll come back to this introductions in a second. I want to talk to you a little bit about the daily structure of the class first. So usually the way this course is going to work, so you're going to do a reading assignment from our textbook before class. So we are using a totally free online textbook, so don't panic, there's nothing you forgot to buy. And this here is a link to what you should be reading. So to our full schedule, the topics we'll go over in class, the relevant page numbers in your textbook. If you have it open in a web browser instead of as a PDF, those page numbers don't always appear. So we also have the section titles for the sections we're going over. Sometimes it'll say only read these specific blocks. So those specific paragraphs will be labeled. Sometimes it'll tell you to skip things, right? So for next time, we're going to be talking about chemistry. You don't need to read about atomic number, or mass, or isotopes. There are a lot of things that we're going to skip over in terms of chemistry because hopefully you have all either already completed chemistry or are currently enrolled in a chemistry course. Okay, so that's that. And I will go through the course links in a minute as well. Okay, so our usual structure is you're going to read before class. You're going to take an individual quiz on Brightspace before class that is just based on those reading assignments. So it'll be on this topic. It's not timed. You can and should have your textbook open. That's not cheating. That is the full intention. They're quizzes because Brightspace labels them as such, but really you can think of them more as kind of homework assignments. That's the kind of level of stress you should be putting into it, right? Kind of like a homework assignment. Okay, so you'll do that before class. And then every class, except for this one, is going to begin with a team quiz. So after today, I'm going to split you into groups because this class is largely going to be based on working through problem sets and case studies together. With a class this big, I'm sure you're in lots of Zoom classes, right? So holding a class discussion with 40 people trying to talk at once, or 40 people not wanting to talk in front of 40 people, is a little difficult. So we're going to break into smaller groups to work through case studies and problem sets. So we're going to have teams. So in those teams before class, or at the beginning of class, you'll take a quiz. That quiz will be the same as the individual quiz or homework assignment you did based on the text beforehand. So the purpose of the team quiz is, one, to get a second shot at the material if you got it wrong the first time around. And second, to have an opportunity to kind of discuss with your classmates. They can be really useful for kind of scaffolding your learning. So they may have useful tips for where they found the information or be able to help explain those questions, although I am always available as well. Then I'll talk for a little bit. There will be a short lecture portion. I know nobody likes to come to a class where they have to do everything on their own. So I will talk, I'll present some material, and then we'll dive into that material and applying it in our activity, case study, or problem set. At the end of every week, we're going to do a short discussion post. And this is just to kind of reflect on your learning, reflect on how the class is going. There'll be different prompts every time, but they'll be more thinking about yourself and your experience in the course. I'm not going to require specific numbers of responses to classmates or anything like that. Okay, and those will be due the following Monday, and that's how the class is going to work. Okay. Let's take a quick tour around Brightspace, because I know one of the things that's sometimes difficult is I hear different professors structure things differently. So we're going to just walk through where you can find everything here. Okay, so this is our Brightspace, our course home. And the way I would like you to access things is through this content tab. The reason for that is that we're going to be splitting everything out by week. Sorry, these dates are copied from the fall session. I'll make sure all the future weeks are updated with the correct dates. Everything for week 1 is ready to go. So if you go through the content, it'll have these specific assignments broken out by week. So that'll keep you kind of on task what assignments need to be done when. They will also be appearing in the course schedule. I believe the course schedule, this calendar gives you guys reminders to your email if you have notifications set up. So that's good. But in general, we want to access things through this weekly calendar. Okay. So here you can see an assignment that we're going to be doing today in class. We're going to be doing like an example problem set. So you can practice how to copy the documents, practice some skills on how you would work together with teammates. We're just going to randomize team members for today, but I will think a little more critically about who should be grouped together in the future. So we're going to practice skills on how we would work on a problem set. No quiz today. And then afterwards, you can see that by tomorrow, I'd like you to complete an introduction survey so that I have a little more information about you so that I can put you into good teams that you'll work well with. Hopefully won't hate each other, right? Make some friends. And you'll have a short review quiz on chemistry to complete for Friday. Okay. So all our weeks are going to look like that. And so you can see through this week, Friday, we're going to cover chemistry. So here is Friday's in-class problem set. Friday's team quiz will open up at the beginning of Friday as well. So those will all be completed in class. So the only things that you need to worry about completing on your own before class are these individual quizzes. So if it says team or if it says problem set, you don't need to do that until we're in class. Yeah. I have a lot of things linked here under the course resources. So at the top, we have our Zoom link for class. But here from this course resources, you can also access the course schedule. So that was the course schedule. I brought up also linked in the intro slides. I'm going to go back to the course resources. We have our textbook directly linked. So here we have our textbook, biology textbook, online, free. You can highlight it and add notes, online, free. And here we have a folder that links all the slides, so all the PowerPoint slides for content that we'll be covering. And as we go, as you test these links, please let me know if anything either isn't visible to you or for some reason you don't have access. I went and tested all this stuff for the first week and I think it's good to go, but you know, always best to double check everything. So here are all the slides that we'll be covering for this course. I don't recommend you get too far ahead because I will skim through and make sure that there's nothing we want to delete, but they're all here and you can look a little bit ahead. So you get to that from the course resources. Okay. Next thing we have on this list are our learning objectives. So your learning objectives are numbered, and basically you want to treat these as a study guide for the unit. This course is super short, so we're gonna have a first unit and a second unit. So that's why you see links to 2 sets of learning objectives. So this first one, learning objectives for exam 1, second, learning objectives for exam 2. They're broken out by day. So you can see that today on day 1, this course is super short, so we are actually going to cover a little bit of material. We're going to just cover what a prokaryote, what a eukaryote is. Those are 2 of our types of cells. So then on Friday, we'll cover some chemistry, specifically these topics. Biological macromolecules will take 2 days. These are our topics, and so on. Okay. So the way I want you to use these learning objectives is really, ideally, come back to them all the time. Right? So the learning objectives list out everything you need to know. If it is not in a learning objective, you do not need to know it. So it can be very helpful to have those learning objectives open or have them printed out as you're reading the textbook, as you're reading those reading assignments. Because if you get to a section, say, right, said you don't need to know atomic number, mass stuff for chemistry, right? You look at this chemical foundations learning objectives, you notice atomic mass, atomic number is nowhere in these learning objectives. So that's a second way you would know you can skip that section in the book. Okay. So have them open before you read the text. Simultaneously, these are telling you what you should be focused on, right? So you should be focused on understanding differences between different types of bonds, ionic, covalent, hydrogen. So this is a good way to organize your notes by learning objectives, so by topic that you need to know. Looking back at them after class and adding in anything we talked about, either in those groups while working through problem sets or during the little mini lectures is very helpful. And then you can also use them to quiz yourself or each other after class. A note here at the bottom, this is really everything you need to know for this class, which means that some of these learning objectives should be things that you're learning not from the me talking portion of class, but from the problem sets or potentially from the textbook and quizzes. All of these things are going together. Okay, so not every learning objective is going to have a very specific PowerPoint slide tied to it. Okay, going to continue walking us through the Brightspace, just to make sure that we understand what's going on. So for office hours, if you click on this blue title, this takes you to an appointment calendar where you can sign up for my office hours. And they're sporadically throughout the week. I try to make sure I have some earlier in the day, some later in the day. I have some in the evening sometimes. I know everybody's schedule is pretty weird this year. Okay. And those office hours will have a location link in the invite, but if you can't find it, here is my Zoom link. So this is where the meeting will happen. Okay. So I said that I was, oh yeah. So I am currently live streaming this class session. In future, after every class, I will add the YouTube video from class to this lecture recordings. Right now this playlist is pretty empty, but all the future videos will appear here under our lecture video playlist. If you want to get a jump ahead on the material, maybe you don't love reading textbooks, another place you can check for resources as you're doing this quizzes is to look at old versions of this course. So this old YouTube videos link is to a playlist of a previous session of this course. So some things will be a little different, but cell biology hasn't really changed since last year. So the content that you need should be there. There may just be a little extra in terms of what we're covering. We've cut a couple topics for time. Okay, so yep, these things are also linked in the syllabus. I tried to put links everywhere that I thought you might go to look for links. So you can find the syllabus under the syllabus. This textbook is just a link to that same textbook, the Biology 2E. This links us to that schedule we already opened up here. Okay. This is linking to my office hours. Here's another Zoom link for my office hours, etc. Okay. Questions about where to find stuff in the Brightspace course as of right now? I know you haven't tried clicking through too many things, but any immediate confusion? Okay, I'm not hearing anything. And if at any point I say I'm not hearing things and you are trying to talk, please just type in the chat. My speakers were a little bit on the fritz in my last class session. I think I've got them working again, but in case I don't, using the chat is always okay. Okay. So we're going to do some quick introductions in breakout rooms instead of in this big 40-person lecture. So what I want you to do in the breakout rooms that I'm about to put you in, introduce yourselves to each other, basic stuff, right? Your name, what major you are, things like that, a fun fact about you, or not so fun fact, any fact, your favorite food, I don't care. What I do want you to find out is one thing that your group shares in common. So not we're all in bio the cell, but I don't know, maybe you all like the same sports team or something. I want you to just do a little socializing, right? Do a little chatting, and then we'll come back together. I'm going to talk a little bit about our content, and then we're going to work on the problem set. Ooh. Sorry, I have old breakout rooms set up from our previous class. Oh gosh, we do not want that. Okay. Sorry about that. I didn't realize that setting was going to be saved. You know what? It's fine. All right, gotta click on all your names. So you can be thinking about what you're going to say in your introduction. I will be breaking you into these same groups for the problem set we're going to do at the end of class. So if you guys get super bored, you can open that link as well. Okay.
I just have a quick question about the readings.
Yes, what's up?
So I saw when you pulled up the syllabus, I guess I just totally like I noticed that we didn't have to buy a book, so I just like didn't really think to work ahead. But there was like 2 readings that are due. Was that like for today that were supposed to be done?
So there are page numbers that go with the topics we're going to go over today. It's fine if you didn't read them yet, but as you're studying, you can use those as a reference. So like when it comes around to our exam, which our first exam is on February 12th, make sure you've gone ahead and taken a look at that.
Okay, so then for Friday, read the pages that you. Okay.
For Friday, go over this stuff. Yeah.
Thank you.
Anybody else have questions? That was a great question.
Thanks. I do have a question.
Mm-hmm.
So the quizzes, how much time do we have? And like, do you know like the normal number of questions?
Sure. The quizzes for when you're taking them individually, they're untimed. So you can have as much time as you want on those. When you take them as a group in class, they're going to be 10 minutes. So as you're taking them individually, think about which ones you want to talk through with your group specifically. Sorry, you had a second part to your question, which was how many questions? There, I believe 7 questions, most of them like 5 to 7. They're pretty short.
And then the exam, how will that work? Like, do we take it in class or?
Yeah, we're going to do the exam in class, but still like over Zoom. So that'll be a mix of multiple choice questions and some short answer questions, because since this class is based a lot on case studies and working through problem sets, those are kind of easier to apply to similar types of questions in short answer type things. Anybody else? These are great questions. Okay, I gotta hit the ground running because 8 weeks is not a long time to cover everything about a cell, let me tell you. Okay, so we're studying life here. Really, we're studying the basics of life. So we're studying cells, and you're also studying the basics on your own before class, after class, and in class, we're trying to focus mainly on applying things. Okay. We saw these upcoming events on the Brightspace, but here they are typed out for you as well. So by the end of the workday tomorrow, I'd like you to fill out the introduction survey. I can see a bunch of you have already done it, so that's great. Only I'm going to view your responses, so don't feel embarrassed. One of the questions is, how do you generally feel about group work? I know some people hate group work, some people love group work, but I want to get a good sense of what your concerns are so that I can pair you with people who you will hopefully fit well with in a group. So if you're the kind of person who feels like you're always taking on the work, I want to know that. If you feel like you're someone who has trouble speaking up or feels like people are rushing too far ahead, I'd also like to know that, because I know those are kind of 2 sticking points people tend to have. Okay, so in class, the end of class, we're going to get to this careers problem set. And for Friday, you're doing quiz 1. In class, we'll do a problem set. Monday, you're doing a quiz before class. In class, we'll do a problem set, and so on. This check-in survey is going to go up on Friday, but won't be due until after class on Monday. So you'll have the weekend and then Monday to do that first discussion post. Okay. I will introduce Brooklyn. I don't think Brooklyn is in the room right now. Brooklyn is our TA and SI leader. No, I don't think she's here. Yeah, Brooklyn is great. She's a senior. She's an ex-phys major. She's going to be a physical therapist. She took this course several years ago, and she's been the SI leader for a number of years. SI is helpful for reviewing class material. Brooklyn's good at explaining things and good at communicating what people are confused about from you guys to me, if you don't want to come to me directly, although I hope you will. I'm a pretty friendly person, and locked away in isolation, you know, I get lonely. So feel free to reach out to me or to Brooklyn. Lots of places you can go to for help for this class. SI is going to be 8 to 10 in the morning on Tuesdays, 4 to 6 in the evening on Thursdays. So you can go either of those times. And the link is here on this slide. In addition to going to SI to ask questions, either about the problem sets, about things you read in the reading that you don't understand, things I covered in lecture that you don't understand. She also has worksheets that review class material. This is helpful for practice, but the worksheets aren't mandatory. I do look at those worksheets sometimes and pull a couple of them for test questions. So they are worth looking through. Okay. She has some advice for what you should do if SI doesn't work in your schedule. Like, you will still have access to those worksheets, even if you can't make it during one of those time slots. Okay, cool. So we're going to come back to this problem set at the end of class. And we already talked through how to navigate Brightspace at warp speed. But I want to talk a little bit about cells because we're halfway through the class period and we haven't really talked about them. Okay. So one of our kind of fundamental topics for this first unit, and really maybe the class as a whole, is we want to understand what cells are, and we want to understand where they come from. Now, those sound sometimes like pretty philosophical questions, but we actually have some quite concrete answers. Right? We're going to be talking about cells from their chemical building blocks as they build from atoms into macromolecules, and then how those molecules are organized into cells. And then we're also going to talk about how cells replicate. So cells are going to come from other cells. We'll talk about how that happens. Okay. So the basics of the answers to these two questions, right? What are cells and where do they come from? Are something called the cell theory. So a tenet is like a rule. So there are 3 tenets of cell theory. And here they are. So the first is that all living organisms are made of one or more cells. So if we say something is alive, it has at least one cell. And if we're talking about something that doesn't have cells, then it's not alive. So this has interesting applications for your future potentially, because, for example, viruses don't have cells. So viruses are often in this iffy gray zone where they're not really actually alive. They're not technically living organisms. Okay, because every living organism is made of one or more cells. Second tenet is that the cell is the basic unit of structure in living organisms. So when we talk about what organisms do and how they function, our basic unit that we look at is the cell. We talk about what the cells do, how the cells function, and how they lead to the behaviors and the structures that living organisms have. So basic unit of structure is a cell, because all living organisms are made of cells. Those two things kind of play off each other. Our third tenet is that cells arise from pre-existing cells. They don't just pop out of the air out of nowhere. Cells replicate through reproduction to create more cells. Okay. That was really revolutionary when they discovered that, but you know, now a little less revolutionary, right? Hopefully we all got that sometime in high school, right? We went through mitosis and meiosis in high school. Probably we're going to go through it again in this class, but not for a little while. Okay, so we know cells are the building blocks of living organisms. All living organisms have cells. But when we look at our cells in specific types of cells, specific named cells, we're going to see that all cells are one of 2 types. They're either going to be prokaryotic or they're going to be eukaryotic. If you look at an organism, we're not going to see a mix of these types. These aren't different types of tissues. These are fundamentally different types of cells. So prokaryotic cells are what we're going to find in organisms like bacteria. Not only bacteria, but that's one of our big classes of organisms that are made up of prokaryotic cells. Eukaryotic cells are going to be slightly more complex. So if we look at a eukaryotic cell, we're going to see it contains more parts. Eukaryotic cells are sometimes going to be single-celled organisms, but they're also going to be the type of cell we see building together into multicellular organisms. So if you're looking at a human, for example, it's going to be made of eukaryotic cells. Humans are eukaryotes. Okay. So there are really important differences between prokaryotic and eukaryotic cells, apart from what type of organism is involved. If we look at the cell itself, we're going to see that they look quite different. Nobody needs to tell you you're looking at a bacteria for you to be able to recognize a prokaryote. You can recognize it from the qualities you could physically see under a microscope. Okay. So the main difference, so the most important difference, I do want you to know lots of different differences between prokaryotes and eukaryotes, but the most important one is whether it has a nucleus or not. So in prokaryotes, you are not going to see a nucleus. In a eukaryote, you are going to see a nucleus. So a nucleus, we'll take a look at what that looks like in a second. And a nucleus is a membrane-bound area that contains genetic information. So it's only present in eukaryotes, never present in prokaryotes. Another difference that we'll notice is size. So prokaryotes are very small, eukaryotes are larger. We'll also see that prokaryotes don't have organelles while eukaryotes do. So things like mitochondria, for example, you will not find them in a prokaryote, you will in a eukaryote. So those are named organelles, and we'll go through those in a couple weeks of the different organelles. You'll see them in eukaryotes, not in prokaryotes. There are also differences in their DNA. We're not going to delve into that too deeply right now, or really in this course, but you should know that prokaryotic and eukaryotic DNA just look different. It's true, they look different. Okay. So here's an example of a prokaryotic cell. So E. coli bacteria is pretty typical for a prokaryotic cell. So these are some E. coli here that we see here. So they have a cell wall, they have a plasma membrane. We can see that there aren't a lot of structures inside this cell, right? It's pretty small. If we compared this number, this unit of measurement, we'd see it's smaller than a eukaryotic cell. We'll notice that while it does have this kind of central area, it's labeled a nucleoid region. So this is not actually a nucleus. We'll see that nuclei are pretty circular, they're dense, and they have a plasma membrane surrounding them. A nucleoid region is more like kind of just a vague sort of area in the cell where you would find genetic material in a prokaryote specifically. One thing that they do have in common with eukaryotes though is ribosomes. So we're going to talk in the future about how ribosomes take genetic information and turn it into proteins. So prokaryotes do have some similar processes to eukaryotes, and that's one of them. They do still make proteins using ribosomes. Okay. So then we get to our eukaryotes. So many eukaryotes are unicellular organisms. So that's not a difference between prokaryotes and eukaryotes. You could just have one cell, a single cellular organism that's a prokaryote. You can have a single cellular organism that's a eukaryote. Often, those single cell eukaryotes are yeasts. So like the yeast in bread or beer, those are eukaryotic cells, yeasts. So they would have a nucleus, they'd have mitochondria, they have organelles, but they are single-celled organisms. Only eukaryotes and not prokaryotes are going to be found in multicellular and more complex organisms. So we can kind of build up from our simple yeast cells to slightly more complex unicellular eukaryotes. So here we see some examples like a paramecium. You don't need to know specific details about the paramecium. This is provided here for your general interest, in case you're curious about what that is. When we're talking about eukaryotic cells, what we're really going to be talking about is multicellular organisms like us. We're going to be talking about human cells mainly, because this course is largely to prepare you for A&P, so human cells for what you're going to see there. So we're really not going to be focusing on small prokaryotic bacterial cells in this course, but I do want to make sure that you know the difference, know those exist, because another course that this is a prerequisite for is microbiome, which will focus a lot more on those prokaryotic cells. So some of these processes that we go over in this course, like DNA replication, are going to be specific to eukaryotes, and some will be shared by eukaryotes and prokaryotes. And as we get to those, we will mention specifically if we're talking about like, okay, this is happening in eukaryotic cells. Okay. So now we're going to practice a problem set. All right. So this is your practice problem set. We're going to go back. So we're going to do the cell bio in my career problem set. And rather than getting there through this link, I think it's best if you get there through the week 1 in Brightspace. And that's because it's an assignment in Brightspace. So then you'll already have up where you should be uploading this file. Okay, so we're going to break out into groups. What I want you to do, I'm going to open this up. Okay. So you're going to see this problem set here with the instructions specifically. So you'll go through. The data for this was from a survey compiled by Dr. O'Brien, who is an instructor for a different section of this course, that she compiled based on her friends in different fields and how they use cell biology in their jobs. Okay. So what you're going to do in order to complete this problem set is you are going to make a copy of this Google Doc. Only one person in your group has to do this because you're all going to work together on the same problem set. So whoever copies the Google Doc is then going to share the copy with their team members. So you're going to make your own copy, then you click the share button, right? And you'll add the people in your group, send them a link. You're going to complete the 4 questions in the problem set together. I do want to make sure that you each know how to do this process. So I want you to each download the file that you work on together. So you'll complete the 4 questions together. You'll all type in the Google Doc together. So then you're going to download this as a PDF. You're going to just upload it to this Brightspace assignment. And after class, I will give you all credit for having done this. Okay. Questions about how to do this right now before I send you into groups? Another thing you can practice while you're in those breakout rooms, this problem set specifically doesn't ask you to do any drawings, but a lot of our future problem sets will involve making drawings of different cells, drawing arrows for where things are moving. So if you want to practice sharing a whiteboard with each other and drawing together, that can also be helpful if you finish the problem set before we're all ready to come back together. Okay, with that, I'm going to open these rooms back up. So you should see a link to rejoin that breakout room you were in before.